Welcome to my poster presentation for the ISMB ECCB 2021 virtual conference. My name is Nathan Mikhaylenko. I work in the Institute of Medical Informatics and Biometry at Technical University of Dresden, and I will talk about our study, as the title says, Gene Expression Profiling Reveals Distinct Molecular Subgroups of T-cell prolymphocytic leukemia, that was done as a part of International ERANET PLL project. T-cell prolymphocytic leukemia, shortly TPLL, is a rare but highly malignant blood cancer. The common therapy typically leads to a relapse of nearly all patients at a median time of 1 year. The stem cell transplantation is more efficient, but it is still low at around 20%. The TPLL characterized by high count of blood white cells and presence of specific TPLL cells with complex chromosomal aberrations compared to normal healthy T cells. The aim of our work was to gain better understanding of the disease mechanism by looking for different molecular TPLL subgroups. Provided by our colleagues, we had gene expression profiles of TPLL cell samples that were used for the clustering analysis. As this is a rare disease, not many patient samples were available. So we clustered 68 cancer samples together with 10 healthy control. We got 4 clusters and checked them for the robustness by bootstrapping. All control samples were clustered together, here in red. Interestingly, one cancer subgroup, blue, shares a subtree with the control, whereas 2 other cancer subgroups were clustered separately. Further comparison of available survival time showed that surprisingly, patients from SG1 group, this is closer to the control, had worse survival than 2 other patients from 2 other groups. Unfortunately, the number of available samples were not enough to draw a significant result. Nevertheless, this finding motivated us to look further into the difference between these 2 TPLL subgroups. We performed differential gene expression analysis to get genes significantly different for each subgroup compared to the control. By applying the threshold at 5% for adjusted p-value, we got following number of significant genes. In accordance to the clustering structure, SG1 has twice less significant genes than either SG2 or SG3, but all subgroups have more underexpressed genes than overexpressed. Following pathway enrichment analysis showed that the TPLL subgroups have some common core set of genes here as overlapping on Venn diagrams, as well as a set of unique genes. And we can see here that overlapping between SG2 and SG3 is more than genes that have SG1 common with either 2 of the other groups. The similarities and differences can help to understand the common mechanism of this type of cancer, as well as subgroup-specific difference that can help design and target specific treatment. In this example of known cancer-related JAK-STAT signaling pathway, we see, for instance, that pathway oncogene MIC was overexpressed in SG1 and SG3, but not in SG2. Then we learned regulatory network with RegNet package based on gene expression and copy number profiles, and we got here a network of highly connected genes, at least 3 links to other genes that were comprised in 16 models. Most of these genes have been reported to be involved in cancer development, and many of them in leukemia development. We can also say that most of these genes were part of biological processes such as immune response, angiogenesis, cellular respiration or oxidative stress, cell proliferation, apoptosis, migration or invasion, and also there is a model involved in nuclear pore complex, this one. By coloring the network with gene expression changes, characterizing different subgroups, here's the legend, we can notice that each subgroup has specific patterns of expression alteration. For example, this model, EFI3 and EFI2, is overexpressed in SG2 group, but is unchanged in 2 others. And we can say the difference in other models, for example, this one, or Elaine connected genes. We also noticed interesting fact that some of models contain pairs of counterplaying genes, such as FIT3 is a oncogene and FIT2 is tumor suppressor. The same is for these pairs and these pairs. And it was reported before for other type of cancers, and it is speculated that oncogene from this pair can be chosen as a drug target. By suppressing it, we can help the organism to defeat and suppress the cancer. In summary, our study focused on the identification and in-depth characterization of molecular TPLL subgroups. Thus, 3 robust TPLL subgroups were revealed by hierarchical clustering, where SG1 was closer to the healthy controls than SG2 and SG3. Characteristic expression differences between the subgroups exist at the level of individual genes, signaling pathways, and network models. Several genes or pathways could represent potential promising targets for the development of drug-based interventions. This project was funded by TransCan2 program, and at the moment we are preparing the manuscript for the special issue in Frontiers in Oncology. Thank you for your attention, and I'm looking forward to answer your questions on my poster panel.